Hey gang, and welcome to another exciting episode of Zoology Online. And today we're going to be talking about phylum Cnidaria and what all is included in that and how they live and what they do. So most of us know that phylum Cnidaria includes jellyfish, anemones, corals, hydras, and a few others. But the word Cnidaria, it means nettle. And in Latin, the word nettle actually refers to stinging. So one of the things that most people know about most cnidarians is that these guys sting. So it's obviously things that you just kind of want to stay away from for the most part. And we'll talk about why they sting and what they use a little bit later on. So this entire phyla are found in aquatic ecosystems. Now, most of these are found in marine ecosystems, but there are a few that can be found in freshwater. One of the big features of their anatomy is that when we looked at sponges, they didn't have any true tissue. They're just a conglomeration of cells. Here we're going to start seeing our first bit of tissue layers, but they're going to be very simple. So they have two tissue layers, two germ layers. And so because of that, we call them diploblastic. Diplo, the di meaning two, a blastic meaning part of their body and how they develop. So they're going to have essentially an internal side and an external side. But another good feature of all of these is that their symmetry is radial. In other words, they're based off a round shape, essentially. You can see this when you look at jellyfish. You can see it when you look at anemones and hydras as well when you look up close. Another defining feature of their body plan is that these are all going to be acoelomates. And if you remember, a coelom is a body cavity, and these are without a body cavity. They do have, as you see later on, they do have a gastrovascular cavity, but that's not the same as having a body cavity. You need to go back and look at the last chapter if you need to look at the development and body plans to go over the different types of coeloms that things can have. Another big feature of these guys is how they develop. There are two different body styles that these guys can have. Essentially, they can either polyps or they can be medusas. And we have a whole slide dedicated to the difference between those two. We'll talk about the nematocysts, their stinging cells. We'll look a little bit at their gastrovascular cavity, which is also defined in a lot of the worksheets that go along with this. You can see those linked in the modules right under the notes section. If you need to print out any of those diagrams, please do, because labeling those will be part of the exam. And that will definitely help prep you for that. They do just have one opening. So when they go through gastrulation and they form that first little indent into the wall of cells, a second one never appears. So these, we can't really say these are mouth first or butt first. These are simply going to be in their own category. They only have one opening, so they don't qualify to be mouth first or butt first. So they have a mouth/anus because what goes in that hole gets digested. Whatever's left over comes out that same hole. Some of my students used to call this manus. They thought they were being funny, and I told them, quite rightfully corrected them that that word was already taken. That's actually a feature on a bird, so we can't use manus, but it's just mouth/anus. So as we see the phyla increase in complexity, we're going to start seeing some rudimentary systems start to form. And in cnidarians, these two systems have to do with nerves and muscles. Now, the nerve is not, they don't really have a nervous system. They have what we call a nervous net or a neural net. And we'll explain more about that as we go on, but they do have a very rudimentary muscle system to where the coordination of the contractions helps push them through water. As far as being able to go through respiration and get rid of waste, they simply rely on diffusion. So each cell is responsible for its own respiration and excretion. So each cell will take in oxygen and nutrients on its own, and then just through simple diffusion it will get rid of waste products like nitrates and other types of cellular waste. So no systems for that yet, and like we see with most simple animals, that the reproduction could either be sexual or asexual, and in some cases both. So, just any which way you can at this point. So these are a lot of things that we're going to talk about the anemones, about the cnidarians, and then we'll talk about all the different types of cnidarians in each class. So first of all, let's look at the form and the function, specifically with their dimorphism. There are two body styles that they can have. So we have the polyp, which is right here, and we have the medusa. Here's an example of that. So the polyp is essentially what we call the hydroid form, and just know that this is the form where essentially they sit on the bottom and they don't move. So we call these sessile. Their bodies are described as being very tubular, in that it's the shape of a tube, and at the top you'll have the mouth/anus, and surrounding that are generally tentacles. Different species, different number of tentacles, different size of the body, but essentially this is the polyp shape. So the opposite of this, or the other half of this, is the medusa shape, and this is essentially the quintessential jellyfish form. It's where you have a bell-shaped disc that is free floating, and on that bell-shaped disc on the underside there'll be a mouth, and surrounding it from the rim are generally tentacles. Got a few here in this cone jelly, in this box jelly, but obviously lots of jellyfish have lots of tentacles. But in the middle, underneath and in the middle of that, you'll find their mouth/anus. So one is sessile and doesn't move. The other one is free floating. So we have our polyp and our medusa, and most will switch from one to the other at some point during their developmental stages. So now that we know that the two types, the polyp and medusa, we can actually look at their germ layers, them being diploblastic. So their two layers are the ectoderm and the endoderm. So the ectoderm is also called the epidermis, meaning the outer part, and the endoderm is also called the gastrodermis because it is their stomach essentially, and that's simply just the inner layers of cells. In between the two, there's no third layer. We usually think maybe they have mesoderm, but they don't. Again, these are diploblastic, not triploblastic. So they have ectoderm and endoderm, and in between there's a jelly-like substance called mesoglia. And inside the mesoglia, you'll find an elastic type of material. It's essentially the jelly part of the jellyfish, but it acts as a skeleton, just makes it slightly more rigid than the surrounding environment to help them be able to keep their shape. So ectoderm on the outside, endoderm, which is the gastrovascular, or the gastroderm, which is the lining of the gastrovascular cavity, and then there's the mouth and anus. And you can see the same thing on the polyp. Notice though, there's a lot more mesoglia in the medusa than there is in the polyp, which is one of the defining features of some of our classes. So we said that there's not really the simple animals don't usually have a lot of systems, and as we get more complex, we'll start seeing more systems develop. So while they don't have a nervous system, it is important to note that they do have what we consider to be a nerve net or a neural net. And essentially this is just a web system of neurons that stretches throughout their body. Essentially they have one net on the outside in their in the ectoderm, and they have another side on the inner part, the endoderm, or the gastrovascular side. These two neural nets are just responsible for helping them to sense a little bit of their not so much as their surroundings, but mostly things that they bump into. And it's also useful in helping to coordinate the movement of their muscle system. But it's very important to know that these do not have brains. There is no brain. That's why it's not a nervous system. There's not different organs combining together to make the neural net. It's just a neural net, so there is no brain. Now some cnidarians do have what we call a raphallium, which is a sensory cell. Essentially, we consider it to be an eye. It helps pick up on light and dark, and it also can help determine what's up from down, so that way they can orient themselves a little better. Now, not all species have this, or not all the classes have this, but some do, and some have it a little bit more strongly than others. So when we look at their muscle system, essentially we see that they have there's two different types of muscles that they have. They have the epithelial muscles, which are found in both of the germ layers, the inside and the outside. And this is essentially, it helps with movement. There's just little myofilaments, which are the basic parts of a muscle cell, and that's what helps do the undulation and the motion that we see here. Now, in the mesoglia, in that jelly-like substance between, we see striated muscle cells. Now, these are very similar to the striated muscles we see in some more higher developed organisms, but they're not the same, but they're similar. But they are definitely striated muscles, and these also will definitely help contribute to the motion of an animal, whether that be between the epithelial muscles and the striated muscles. They can do things like move and swim through the water column, or they can actually retract a tentacle to help pull up prey that they've caught. So any of that movement is done by one or the other of these types of muscles, or the muscles moving together. So other than that, they do have a specialized type of cell, or one of the specialized types of cells at least, is they have gland cells. So once they capture prey and they pull it inside of their gastrovascular cavity, these gland cells essentially release digestive fluids to help break down and dissolve the organism, and then the juices and the nutrients that were digested, broken down, and absorbed simply get absorbed, and through diffusion will spread through the rest of the organism. All right. Now let's talk about the nematocysts. This is a cool part. So this is another feature of their anatomy that pretty much all cnidarians share. And the nematocyst, a fun word to say first of all, this is essentially the stinging cell, right? So here we have a good little diagram of what it would look like. So here we have the cnidocyte. You can just leave the C off of these words and just say the rest of it. It's the cnidocyte. That's the cell that contains a capsule, and that capsule is the nematocyst. That capsule has a covering over the top of it called an operculum, and it's just the lid that covers the part where the barb and the harpoon are located. Sticking out from that is a cnidocyl. A cnidocyl is simply a trigger that, once disturbed, will pop open the operculum, and it releases the pressure that's inside, because these usually nematocysts have a high amount of pressure inside of them, which allows the barb to shoot out like a spear and be able to sting and deliver the venom to their attacker or to their prey. Now keep in mind that the cnidocyl, this trigger, is not found in Anthrozoa. So that's the one class that actually doesn't have a trigger. They have a little bit more control over their nematocysts. They don't have to wait for an external stimuli. They can actually trigger the release of the barb themselves. So now, when we look at the nematocyst, the stinging cell, you can see that there's this thread that wraps around. There's a venom sac that's located on the inside, and once it's usually sprung, that's it. It's sprung, and that thread will, the barb and the thread will attach to the attacker or to its prey, and continue to deliver venom as long as it's attached. Now, the barbs themselves come in a variety of different shapes and features, and that's one of the things that we use to help determine the differences between different species of jellyfish that otherwise look the same. Or not just jellyfish, but any type of cnidarian. Hey, it looks similar. Well, let's look at the barbs. Well, you spend a lot of time under a microscope doing that, looking at these very, very small features, but that can definitely help determine one species of similar jellyfish from another. So, and by the way, if you've ever seen season 1 of Stranger Things, you'll get the joke on this.